Now we're going to take a look at how this Now we're going to take a look at how this process actually is carried out. This is done by important photosystems. There are two photosystems within this process. We call them photosystem 2 or PS2 and PS1. An important thing to note is that these photosystems were named PS2 and PS1 with respect to the order in which they were discovered. However, we will see that the order in which they act is different. Photosystem 2 is actually the first photosystem in the photosynthetic process. So please do not get confused between the way they are named and their position in the thylakoid membrane. There is an important light harvesting complex that is present within the membrane of the thylakoid. In these light harvesting complexes, photons of light are going to be used to excite an electron of chlorophyll to a higher energy state. We call this photoexcitation. This excitement is going to be passed around, and eventually these electrons are going to be passed to a specialized chlorophyll pigment of chlorophyll A called P680, which is present in photosystem 2. P680 is termed P680 because it has the maximum absorption level at 680 nanometers. That refers to the maximum wavelength of absorption. These electrons are this energy is then going to be used to excite an electron from P680. Once this electron is excited, it is going to jump to the primary electron acceptor. The primary electron acceptor will only accept an electron if it is in the proper high energy state. If it is not in the proper high energy state, it will simply remain in P680 until enough energy is transferred to it. Once the primary electron acceptor accepts the high energy electron, it will then be passed to the other photocenter. Once these electrons leave photosystem 2, there is going to be an electron deficit. So in order to replace these lost electrons, P680 will be stealing additional electrons from water. This is called the splitting of the water molecule. When the water molecule is split, this will create oxygen as a byproduct. These low energy electrons will then sit in P680 until another photon can be used to transfer more energy to these electrons. This process will occur over and over again. Once the electrons are energized to a high enough energy state, they will leave the primary electron acceptor of photosystem 2. These high energy electrons will then be passed to photosystem 1 through an electron transport chain. In this case, this is called a cytochrome complex. So once again, we have the appearance of an electron transport chain. I would like to simply remind you, an electron transport chain is simply a grouping of proteins that allow for electrons to pass through them. In this case, we have it existing between photosystem 2 and photosystem 1, and in this case, it is called the cytochrome complex. Once again, we're going to see a common occurrence where some of the energy from these electrons are going to be used to make ATP. As a result, these electrons are going to lose some of their energy as they pass through this electron transport chain. This means when they reach photosystem 1, they're going to be in a lower energy state. We then need to re-excite these electrons to a high energy state in order for them to be accepted once again by the primary electron acceptor. In this case, we're going to have another photon of light that will be used. This energy from this photon is going to pass through the various chlorophyll pigments. Eventually, this energy is going to get passed onto a specialized pair of chlorophyll molecules once again. In this case, in this photosystem, we refer to them, that specialized pigment, as P700. Once again, this is simply referring to the maximum wavelength absorption of 700 nanometers in this chlorophyll pigment. The energy is going to be transferred to these electrons. They're going to jump to a higher energy state, and once they have enough energy, they're going to be delivered to the primary electron acceptor. Provided they have enough energy, the primary electron acceptor will then allow them to leave this photosystem, and they will enter into another electron transport chain. However, in this case, we have ferredoxin, which is considered the electron transport chain that receives those high energy electrons from photosystem 1. Ferredoxin is responsible for passing on these high energy electrons to NADP+, thus reducing NAD+, and forming NADPH. We are going to take a look at that overall process. Here we have the overall layout of a photosystem. We have a photon of light that can come in, and that energy from that photon is going to be transferred between those chlorophyll pigments. Within the center of this light harvesting complex, we have the reaction center complex, which contains the specialized chlorophyll pigment. Depending on the photosystem, that will be P680 in photosystem 2, or it can be P700 in photosystem 1. Once that energy is transferred to those electrons, they will enter into a high energy state. They will then be accepted by the primary electron acceptor. Once the primary electron acceptor has taken in these high energy electrons, and considering they're high energy enough, they will then be passed on to the respective electron transport chain. This slide gives an overview of the two different photosystems and their respective electron transport chain. So let's begin with the first photon of light entering in photosystem 2. That energy is going to be passed around those different chlorophyll pigments, and it is then going to be transferred to the electrons at P680. These electrons will then leave P680, be accepted by the primary electron acceptor, and then be passed to the electron transport chain that is located between PS2 and PS1. We call this the cytochrome complex. The cytochrome complex consists of various types of proteins and also consists of plastoquinone and plastocyanin, PQ and PC for short. Together, these make up the electron transport chain that allows for those electrons to pass between photosystem 2 and photosystem 1. Remember that once these electrons leave photosystem 2, we have an electron deficit. This is where we need to replace those electrons using water. So P680 is a very specialized chlorophyll pigment, and it is a very strong oxidizing agent. So much so that it can actually strip away electrons from oxygen, which is not a very easy thing to do given oxygen has such a high electronegativity value. So P680 is considered to be an even stronger oxidizing agent than oxygen. When it strips away these electrons from water, it will split apart the water molecule. As a result, we have the formation of oxygen, which we can see in the diagram in red. We will also have the production of protons, which will be important when we take a look at the cytochrome complex and what it does. So these high energy electrons are going to be passed through this cytochrome complex. Remember, they're high energy electrons passing through an electron transport chain. As we have seen already in respiration, when electrons are passing through an electron transport chain, there can be the ability to pump protons. In this case, these protons are going to be pumped towards the interior of the thylakoid, meaning that they're going to be pumped from the stroma to the thylakoid space. You can see the red arrow with the 4H plus represents the protons that are going to be taken from the stroma. They're going to pass through, and the cytochrome complex will act as a proton pump, and it will pump these protons to the interior of the thylakoid space. Remember, this is active transport, so that means that energy is being used, and that energy is coming from the electrons. Also keep in mind with active transport, we are going against a concentration gradient, so we are going from a low concentration to high concentration. That means that there must be a low concentration of protons in the stroma and a high concentration of protons in the thylakoid space. Once these electrons pass through the cytochrome complex, they are going to be in a lower energy state. This is when they enter into photosystem 1. Because they don't have enough energy, another photon of light is going to come in, and that energy is going to once again be transferred amongst chlorophyll pigments to eventually excite these electrons in the specialized chlorophyll pigment of P700. Once these electrons obtain enough energy and reach a high enough energy state, they will be sent to the primary electron acceptor of photosystem 1. Provided they have enough energy, they will then be passed to the next electron transport chain, in this case ferredoxin. Ferredoxin will then allow for those electrons to then be transferred to make NADPH. And in this case, we have NADP plus reductase, which is simply an enzyme that will help catalyze that reaction. It will take NADP plus, it will also take protons, and it will help take in those electrons to reduce it to form NADPH. So we've seen how protons can be pumped into the thylakoid space. We've also seen how these electrons will then be used to eventually be re-energized and then placed into an NADPH molecule. If you take a look at the bottom of this diagram, we have an enzyme which we've already seen in the process of respiration. Here in photosynthesis, we also have the incorporation of ATP synthase. ATP synthase simply does what it does in respiration. It will take advantage of the proton gradient and it will use that to make ATP molecules. However, notice that the orientation of ATP synthase has changed, which means that here we've got a high concentration of protons in the thylakoid space. Those protons will flow from high to low, because remember, this is passive transport. So these protons are going to flow from the thylakoid space into the stroma. As they flow from high to low, this is going to turn the turbine of ATP synthase, which is then going to harvest that energy from those protons flowing through to produce an ATP molecule. Note that NADPH and ATP are both being produced in the stroma of the chloroplast. This is very important because the Calvin cycle occurs in the stroma. This means that the products of the light reactions are going to be both produced in the stroma so they can be readily used by the Calvin cycle. This is a series of animations that will take us through, once again, that process. This begins, once again, with photosystem 2. Remember, photosystem 2 has a specialized chlorophyll pigment, P680, which is a strong oxidizing agent. We have the presence of a light harvesting complex that consists of various chlorophyll pigments that will help absorb the energy from the sun and transfer it to those electrons being housed in P680. Once those electrons are energized enough, you will then get them being delivered to the primary electron acceptor. From here, they would then go on to the electron transport chain. Note that once we have the electrons leaving P680, there is an electron deficit. This needs to be replaced with new electrons. These new electrons come from the splitting of water. So here we see a water molecule being split. This will produce protons, and it will also produce oxygen as a byproduct. These electrons, once energized, will then get passed to the electron transport chain, consisting of plastoquinone, the cytochrome complex, and also plastocyanin. Once these electrons are passed through the electron transport chain, some of that energy is going to be used to pump protons to indirectly make ATP molecules. When they reach photosystem 1, they are going to be in a lower energy state. This means that they need to be re-energized. Where does that energy come from? It comes from another light photon. So the similar process is going to occur. Light is going to hit the photosystem. This light energy is going to be harvested amongst the chlorophyll pigments. This energy will then be transferred to these electrons being held, in this case, at P700. Once these electrons are energized enough, they will then be delivered to the primary electron acceptor. Once they have enough energy, they will then be passed on to the next electron transport chain. In this case, the electron transport chain is ferredoxin. Ferredoxin will transfer these electrons to NADPH through the use of NADP+ reductase. This diagram here illustrates this process and flow of electrons from the photon to the electrons through the electron transport chain, where some of that energy is going to be used to make ATP molecules. These electrons will then be re-energized to a higher energy state and then eventually be delivered to form NADPH. We refer to this pathway as the linear flow of electrons. However, although this creates ATP, and NADPH, it does not create enough ATP to carry out the dark reactions. We will also see that in addition to this linear flow of electrons, there exists a cyclic flow to electrons in the photosynthetic pathway. This only occurs in photosystem 1, which means that there doesn't need to be any additional splitting of water and no additional oxygen is going to be produced by the cyclic flow of electrons. We're simply going to be recycling these electrons through the photosystem in order to produce a bit more ATP. When electrons follow the cyclic pathway, when they leave cytochrome C, they will enter into the P700 of the photosystem 1. They will go to the primary electron acceptor, they will go to ferredoxin. However, in the cyclic pathway, they will be recycled back to cytochrome C instead of being delivered to form NADPH. They will continue to pass through the cytochrome C complex. As they pass through the cytochrome C complex, remember, protons are going to be pumped from the stroma into the thylakoid space, and this can be used to generate additional ATP. Why do plants need to do this? This is because if we take a look at the Calvin cycle, the Calvin cycle consumes a little bit more ATP than it does NADPH. If you take a look at linear flow, you get relatively the same amounts of NADPH and ATP being produced. This is not enough to keep the Calvin cycle going. The Calvin cycle needs a little bit more ATP. So this cyclic flow can help to accommodate that ATP demand. If we take a look at ATP production in photosynthesis, although the electrons follow a different pathway, how they are used to make ATP is similar whether you're dealing with respiration or photosynthesis, meaning that we have an electron transport chain that works in pumping hydrogen atoms or hydrogen ions, protons, from the stroma into the thylakoid space. They're doing this to build up a proton gradient. Once this proton gradient is established, ATP synthase can take advantage of it to allow for these protons to flow back down their gradient to make ATP molecules. Take a look at this diagram and look at the flow of electrons and the proton flow. So as electrons pass through cytochrome C, we have protons being pumped, once again, from the stroma into the thylakoid space, creating a high proton concentration in the thylakoid space. ATP synthase can take advantage of that, allow for these protons to passively flow from the thylakoid space back into the stroma, and in doing so has the ability to make ATP molecules. The Calvin cycle is important because it uses ATP and NADPH to synthesize glucose, specifically sugar precursor molecules in the form of G3P, or glyceraldehyde 3-phosphate. These are called the dark reactions because no photons are required. The Calvin cycle uses both products, ATP and NADPH, from the light reactions. The Calvin cycle is similar to the citric acid cycle, but in this case, it is considered to be anabolic, not catabolic, as we've seen in the citric acid cycle. In the Calvin cycle, we are going to be building organic molecules. There are 3 phases to the carbon cycle: carbon fixation, carbon reduction, and regeneration. In carbon fixation, we have the important enzyme rubisco. Rubisco is one of the most abundant proteins in chloroplasts and quite possibly the most abundant protein on Earth. Rubisco is responsible for catalyzing the reaction of adding carbon dioxide to RUBP, or ribulose 1,5-bisphosphate. This is a 5-carbon molecule and is considered to be the carbon acceptor. It will take in that carbon from carbon dioxide to make a 6-carbon molecule. In carbon reduction, we will see that the electrons from NADPH will be used to reduce carbon. Finally, we will have the regeneration phase, where some of these precursor sugar molecules, G3P or glyceraldehyde 3-phosphate, will be used to regenerate your 5-carbon carbon acceptor molecule of 1,5-ribulose 1,5-bisphosphate, or RUBP. This diagram here outlines the Calvin cycle. Once again, I don't want you to get too involved in understanding the overall structure of these compounds. However, I would like you to understand what is going on with the carbons and some important carbon structures such as RUBP and G3P. So at the beginning of this cycle, we have RUBP. The carbons in that molecule are represented by the 5 gray circles. We can see that RUBP has 5 carbons associated with that molecule. We will also have the addition of carbon dioxide. When carbon dioxide joins to RUBP, once again this is catalyzed by the enzyme rubisco, we have the creation of a 6-carbon molecule. This 6-carbon molecule is considered to be very unstable. Almost immediately after it's produced, it is broken down into a 3-carbon molecule, or 3-phosphoglycerate. Now, keep in mind that for every 3 molecules of RUBP, you will be adding 3 molecules of carbon dioxide to then make 6 of these 3-carbon molecules. This simply represents the overall balanced equation. What I would like you to understand is that you have a 5-carbon molecule that is going to have carbon dioxide added to it. This is that carbon fixation. We're making a carbon-carbon bond. We're going from a 5-carbon molecule to an unstable 6-carbon molecule. Once that 6-carbon molecule is formed, it will then break down into a 3-carbon molecule. This is considered to be phase 1, or carbon fixation. We now have to enter into the second phase, which is carbon reduction. This is the part where these electrons are going to be added onto the 3-carbon molecule. In this phase, we will utilize the ATP and NADPH from the light reactions. We can see that there's going to be some of the ATP used because we're going to need those phosphate groups and energy, once again, to be invested into those molecules. And we will also be using those high energy electrons from NADPH. This will lead to the formation of G3P. Remember, G3P is that important molecule that we also saw in the process of glycolysis. G3P was that important intermediary molecule. Glucose was eventually used to form two molecules of pyruvate, and G3P was that phosphorylated intermediate. So that's that one molecule that links those two processes. So once G3P, or glyceraldehyde 3-phosphate, is produced, we can see there's 3 carbons in it, and there's also going to be a phosphate group attached to it. These G3P molecules can choose one of 2 pathways. Some of these G3P molecules are going to be kicked out of the Calvin cycle and used to form sugars. This is why I say that the Calvin cycle overall is used to make the precursor to sugar molecules. These G3P molecules will go on to form glucose, starch, cellulose, etc. However, some of these G3P molecules will stay in the cycle because they have to be used to enter into phase 3. We need to regenerate that important 5-carbon molecule of RUBP. So this is what we see in phase 3. Some of those G3P molecules will be used to form that 5-carbon molecule. This uses the additional ATP that is required through the cyclic electron flow. So we can see overall, we've got the input of carbon dioxide coming in to fix the carbon. We have ribulose 1,5-bisphosphate being our carbon acceptor. Rubisco is important in helping catalyze that reaction to form that carbon-carbon bond and to create that 6-carbon sugar. That 6-carbon sugar breaks down, it's very unstable, breaks down quickly into 3-phosphoglycerate. We then have carbon reduction, where energy is used as well as electrons to reduce the carbon in those 3 carbon molecules. We get the formation of G3P, glyceraldehyde 3-phosphate. Some of those will be precursors to various sugars, and some of those will enter into the regeneration phase, where we regenerate our 5-carbon molecule of RUBP. This utilizes that extra energy that is needed through electron cycling. This diagram here gives you an overview of, once again, those 2 processes. So we have light coming into the light reactions. We have water coming in, providing those electrons that need to be energized. So the light energy is going to be transferred from the photons of light through the use of chlorophyll pigments to those electrons being stripped from water. This is going to lead to the production of oxygen. Also in the light reactions, we have the production of ATP and NADPH. Remember that we have cyclic electron flow, so we have additional ATP being produced from the light reactions. ATP and NADPH are important reactants for the Calvin cycle. This provides the energy and electrons for the carbon reduction phase. It also provides the excess energy to regenerate our RUBP molecule. The Calvin cycle will utilize carbon dioxide or inorganic carbon and fix it to that RUBP molecule. We also have the production of our sugar precursor molecule, specifically G3P, which can then be used to form various types of sugars. The process that you have seen in the previous slides represents the common C3 pathway in plant cells. The C3 pathway is typically carried out in mesophyll cells. However, there are some plant adaptations that plants can have that can help them overcome difficulties in carrying out photosynthesis in various types of environments. C4 plants make a 4-carbon intermediate. They also divide up this process between 2 different cells, the mesophyll cells and the bundle sheath cells. This 4-carbon intermediate is produced in the mesophyll cell, and this can then be broken down into a 3-carbon molecule that can then be used by the bundle sheath cells to carry out normal Calvin cycle processes. CAM plants can divide up these processes temporally, meaning they divide them up over time. This means that they are going to be producing their carbon compounds at night. During the day, the normal Calvin cycle will occur, where the precursor sugar molecules will be produced. However, at night, these additional 4-carbon compounds will then be produced. This means that they can devote most of the day into harvesting solar energy, and they can utilize this energy and make it into organic compounds primarily at night. In addition, these C4 plants can utilize another enzyme aside from ribisco to help in the carbon fixation process in mesophyll cells. This enzyme is called PEP carboxylase or phosphoenolpyruvate carboxylase. CAM plants carry out CAM photosynthesis or crussylation acid metabolism. These plants tend to open up their stomata at night in order to minimize water loss. CAM plants are very present in arid environments such as the desert